All right. I think one of my themes this semester has been with all these historical vignettes that I indulge in, is to let you know that you are indeed part of a long history, an exceptionally long history of interest in the human body, of study and dedication to the arts of anatomy and physiology. And so this is what this classroom would have looked like about 400 and some years ago. Not actually radically dissimilar, kind of a little bit more vertiginous there. You didn't get to sit in a desk and write. What you would have seen would have been this multi-story sort of high vaulted ceiling stacked room with these tightly spaced concentric ring balconies. And then at the very bottom of it here would have been a display pedestal. And maybe you'd have a cadaver laid out and there would be some person down there, an anatomist, doing a dissection and narrating that event for those gathered in front of them. And then having a conversation, people being able to ask questions as this person is doing this thing. This is the first place where venous valves were discovered based upon careful dissection. So you really do have to be quite deft with your hands to be able to reveal. It's been 2004 was the last cadaver that I dissected myself. And I certainly was able to reveal a venous valve, but you got to be gentle. You got to be careful to do that. So you could fit 200 people in here and watch this, and they, on living patients, were able to demonstrate the function of venous valves. This is all here in Italy. This is called the theater of Girolamo Fabrizio d'Acquapendente. So in this guy's book, this is an illustration from his book in which he is demonstrating the function of a venous valve. Now, has anybody ever played with their valves like this on the veins in their arm? You can do this. It's sort of fun. Yeah. Here they have this person. They got a tourniquet to make the veins pop out, and he's flexing himself here. And what you can do is take your finger and press it on one of these bulges in the vein, and then clear the blood from that upstream vein. And you'll notice, so he's preventing new blood from coming in, and you'll notice that the blood that's upstream is prevented from flowing backwards to fill the void space by the valve that's there. So you can experiment on your own arm and see that. This was first time venous valves were depicted. And I'll talk about how they work, venous valves work, at the end of the hour, maybe. Another important point, another important data point relevant to our discussion today. So these are some drawings by Anton van Leeuwenhoek, a Dutch person. Was the first person to actually draw blood cells, and you see these very top there, these very sort of simplistic depictions of what he called red corpuscles. He was looking through a simple reflected light non-compound microscope, and the drawing was made in 1695. So he sees the cellularity of the blood, and he's also got some sort of a depiction of the differences in color. So there are all these lighter ones, and then he's showing a darker picture, a darker one in the corner, which presumably would be a white blood cell. Later, about 100 and a little more than 150 years later, 170 years later, this guy Lionel Beale, a Brit, has this drawing that we see here in his book called The Microscope in Medicine, well after the advent of the compound microscope. And a much more nuanced, sophisticated, and kind of sublime picture of red blood. So this blood was not prepared with a fixative of any sort, and we have, what do we have here? So we have cells of different sizes, and we have these cords connecting them. We have these spiky things. We see granules inside some of them. What might that be? If you were to guess, based on what I said earlier today in lab, you guess what that is? Maybe a monocyte. Maybe a monocyte. Large thing with kind of a large thing. What about this little spiky sea urchin there? I think that is. Yeah, probably a platelet. And if you were to guess, this is not anything that you would be able to guess based on what I've said here that much. Maybe, I guess I did mention it in passing. What would you guess these cords, these ropes are? Maybe fibrin. Maybe fibrin. This is blood that's coagulating. This is blood that's clotting. He's taken a blood sample, not put any anticoagulant in it or any fixative. That blood is coagulating. And so we have fibrin that's creating this meshwork in the clotting blood he's looking at. Okay, so to try to help you guys keep your minds organized as you go through this, I've given you different ways. Sometimes in the beginning I'll give you an outline of the material, and another way you could do it is just to go through the slides and try to figure out what the objectives are. So this is another sort of a way to preview material. I've done this a number of different times throughout the semester. So what are the objectives of what I'm going to talk about? This could be one way. Maybe you don't want to take detailed notes, but just glance through the notes and try to make some sense of what the prof is going to talk about before you get to class. So I'm going to talk about the basic function and composition of blood. There's several slides related to that. I'm going to talk about the structure of a red blood cell and hemoglobin. So I'm going to transition from whole blood into the understanding the structure of a single cell. And then I'll talk a little bit about blood typing and how we recycle red blood cells. And finally, hemostasis, the clotting a little bit. I'm not going to go into detail about it because I think I've already said blood clotting is an extremely complicated topic. And then we'll talk a little bit about some pathology, sickle cell anemia, and hemolytic anemia of the newborn. Those are two pretty interesting blood-related diseases. Okay, that's just this section, and I'm going to do that again for different sections of the talk today. So body fluid and body compartments. All right, so your total body water volume is about 40 liters. It's about approximately 10 gallons of water. So 60% of your body weight is water. It's quite a bit. That's broken up into these different portions. Most of it, 25 liters, about 40% of your body weight is inside the cells of your body. So each of the cells, their cytoplasm, the aqueous component of your cytoplasm, that equals about 25 liters of your 40. And then the rest of it is extracellular fluid. 12 liters or 80% of this 20% are interstitial. So that's the fluid in between cells. And then lastly, about 3 liters of water in your plasma volume. So that's your blood. All right. 20%, I'm sorry, 3 liters of plasma volume, just water in your blood. What does it do? Well, of course, it is it's the sort of logistics medium for the body. It's how you transport all kinds of dissolved nutrients, wastes, oxygen, carbon dioxide, various hormones. This is how the white blood cells get around. The blood is a tremendous buffer to the pH of the body, and is also helps to stabilize the osmolarity of the body. So there's osmoregulation in terms of the ions that are dissolved in the blood. Blood is important for hemostasis, or the cessation of the prevention of loss of fluid whenever you get injured someplace. So the blood, we've already talked about platelets and fibrin just a little bit. We'll talk about it more. The blood has all these white blood cells, which are part of your immune system. They'll protect against pathogens and some types of toxin. Importantly, the body is, we talked about 40 liters, 3 liters of which is the blood. That water has a tremendous heat capacity because of all the hydrogen bonds that water is able to form, intermolecular hydrogen bonds. And because of that heat capacity, it has a tremendous ability to stabilize the body temperature. All right. So, specifically, where you localize that blood versus the core versus the peripheral blood. Whether your peripheral blood, you're going to radiate heat, localizing it to the core, you're going to concentrate heat. It helps to regulate body temperature. Okay, so let's look at the fractionation of the blood. So when you fractionate your blood, that's the separation of, you're going to remove some blood and you're going to separate it into its components for some sort of analysis. So I already said that the blood volume itself was 3 liters of water, but the total blood volume with all the formed elements, that is the proteins and cells, etc., is about 7% of your body weight. So there's quite a bit of weight tied up in your blood. In a typical adult male, that is 5 to 6 liters, and in a female, it's 4 to 5 liters. So, I don't know, maybe 60-70% of your blood is water, but then there's quite a bit of other materials in there, which we'll talk about. 2 little definitions first. A person who is hypovolemic has a low blood volume. And a person who is hypervolemic has a high blood volume. A person who is hypervolemic is probably hypertensive, meaning they have high blood pressure. If you are hypovolemic, you are probably, but not necessarily, hypotensive, meaning you have a low blood pressure. There can be they're correlated, but not. They're not the same. They're not synonymous. All right, so we're going to spin down some blood. We're doing some venipuncture here. I'm going to draw a tube of blood, and we're going to spin it down. And when you spin it down, well, first of all, when you draw it, you'll notice that the temperature is a little bit higher than the temperature that you would read with a thermometer. It's a little over 100 degrees. It's very high in viscosity because of all the stuff that's in it. And this is important, that the pH of of your blood needs to stay in a really tight range. So 7.3 to 7.45, 7.35 to 7.45 is the range. If you go outside of that range, you're not going to feel well. You are really not going to feel well. If you go far outside of that range, you will lose consciousness and risk death. So your blood wants to stay in a really narrow pH range. You spin it down, what are you going to see? Well, you'll see this like dense red thing on the bottom. There'll be this white band above it, and then this a lot of clear yellow fluid on the top. So this is plasma. This is what's called the buffy coat. That's all the white blood cells, and then below it is the erythrocyte pack. We're going to look at each of these one at a time, each of these sections. So first, the plasma. What is the plasma? Well, it's 92% of the 92% of it is the water. There are some other solutes there, though. There's primarily proteins that are dissolved, that are not suspended in solution, but that are actually dissolved. So 7% of the weight is proteins, and that is separated across mostly albumin and globulins. So 95% of the proteins in your plasma are either going to be an albumin or one of the globulins. So albumins are what maintain osmotic pressure in the blood, and they can be transport molecules for hormones. Some albumins transport lipids. The globulins are a massive superfamily of proteins that do all kinds of things. They're also involved in transport of hormones or lipids, ions sometimes. But they include the immunoglobulins. So the immunoglobulins are antibodies. And antibodies are the principal form of adaptive immunity in your defense. There's a little bit of fibrinogen. So whenever you see a protein whose name has ogen on the end, it is what's called a proenzyme. And this doesn't mean that it's professional. It just means that it's not actually active yet. It's fibrinogen is going to get enzymatically activated. In fact, it's going to get cleaved. I have a little regulatory tail cut off of it. And once that happens, fibrinogen becomes fibrin. And once you have fibrin, fibrin is active. It will act as, we showed you in that picture from Beal, Lionel Beal, that he had those cables running across all the different blood cells. That's what fibrin does. It forms clots. We'll see. I have a pretty dramatic picture of it. We'll see in a bit. There's regulatory proteins, enzymes, other proenzymes, and hormones, etc. So then there are other smaller solutes, right? Proteins tend to be the big boys. And the solutes can be different electrolytes. These include any of your small elemental cations or anions, for example, sodium, potassium, calcium, magnesium chloride, etc. And then there can be some organic nutrients. This includes things like sugars, glucose, and some small lipids, some individual cholesterol, and any free amino acids that might be in your blood. And then lastly, there are going to be some organic wastes. So organic wastes like urea, or what do I have listed here? Uric acid, creatine can get put out by muscle cells, particularly if you have a muscle wasting or some sort of an injury. Bilirubin is put out by the liver. Ammonia can be a metabolic byproduct of protein metabolism. All right. So now on to the formed elements. So your red blood cell count can be vary with time and definitely varies across the genders. Most men, the typical range for men is around 5 million, where the typical number for women is around 4.5 or a little higher million red blood cell counts per microliter. And your hematocrit is what is called the packed cell volume. So, for example, if we were to take a sample of whole blood, spin it down, your hematocrit, say this is the whole length of it is 100%. Hematocrit would be what percentage of that does this represent? And in men, that is around 50%, and in women, it's around 40%. Those are typical numbers. Your numbers can be high, your numbers can be low. Certainly, a person's numbers are going to vary with time within a person. Different conditions can affect that. So, of the formed elements, these are ones that are in suspension that when you spin it, drop out of suspension. We have all the things that we talked about in lab today: platelets, white blood cells, and the red blood cells. White blood cells are only 1/10 of 1% of the cells that you'll find in your blood. And we said that basophil is 0.5%. So you can imagine that it's like 1 in, what is that? 500,000, is that right? 1 in 500,000 cells is a basophil. Red blood cells have no nucleus, have no organelles of any sort, really. They have no mitochondria, no ribosomes. So because of that, there's a couple consequences. Number one, they have no repair. They're not going to be able to fix themselves. They're kind of disposable cells. We're going to use them for a while, then we're going to recycle them. Next, because there are no mitochondria, their metabolism is entirely anaerobic. All right. So I did. I showed you a little bit in the muscle chapter. I showed you a little bit of central metabolism. Probably a bunch of you have already had some central metabolism. But for the breakdown of a single glucose in anaerobic metabolism, you're only getting 2 ATP, whereas you get 34 ATP out of the same glucose out of aerobic metabolism. So not an efficient way to burn energy. Thankfully, they don't need a lot of energy. The goal of an RBC is to deliver oxygen. And you want to get the cell as small as possible to fit through these tiny little capillaries. And to do that, they basically throw everything overboard that's not necessary. When you look at the passage of red blood cells through a capillary, you notice these things called rouleaux, R-O-U-L-E. Rouleau is simply a stack of red blood cells that have been packed into a narrow capillary for transport. They do this so that they can maximize the number of red blood cells that are filling the volume of a capillary, right? They stack up on top of one another. If they were round spheres, there would be all that wasted space in there. So the shape of a red blood cell has evolved so that they can stack and you can fit a lot of them. So you're wasting minimal volume in the capillary, but you're maximizing surface area to volume in terms of exchange of oxygen off of the red blood cell. It's pretty amazing set of evolutionary pressures that lead to what we have. All right, so any of these red blood cells are full of hemoglobin. I will talk a little bit more about hemoglobin during the respiratory chapter. Unfortunately, I speak a lot more about it in 275. I don't don't really have the time here, but here it is. It's an amazing protein, and it was the first protein that was identified that was subject to what's called allosteria, or the changing of its function based upon the binding state of the ligand that it's supposed to bind. So hemoglobin itself is a tetramer. That means it has quaternary structure, for those of you who've had a little bit of biochemistry. The primary sequence is just the amino acid. Secondary structure are these beta helices. Tertiary structure is the overall fold of a protein, but quaternary structure is how different subunits can come together and create a larger superstructure. So it has this quaternary structure. There are 4 subunits. There's an alpha and a beta subunit, and there's 2 alpha and 2 beta. They're very similar to one another. When you look at any one of these subunits, they all have what's called a heme molecule. And a heme molecule is simply what's called a porphyrin ring with an iron coordinated at the center. A porphyrin ring is everything that's in beige there. And then when it binds iron at the center of what's kind of like a trampoline or a big molecular spider web, it becomes known as the heme molecule. So hemoglobin is a globin protein that has a heme embedded in it. And it is this iron in the heme, this iron that's coordinated by the porphyrin ring, which is going to bind to oxygen and give hemoglobin the oxygen transport capacity that we are familiar with. If you look at a normal male has, maybe 16 grams per deciliter. A deciliter is, like a large shot glass, maybe what's that called? A green tea cup, something like that, maybe a deciliter. So there's about 14 to 18 grams of this protein in that volume. And a female has just a little bit less, lower density. Does anyone want to guess why that might be the case? What particular burden on hemoglobin concentration do adult women have to deal with? Sure that, but not all women have children. Menstruation. It's menstruation. Okay. So blood typing. All right. So what a blood type is simply a recognition or a reflection of different antigens that are going to be expressed on the surface of the red blood cell. All right, there are antigens. Does anyone not know what the word antigen means? Okay, an antigen is you can think of it as a molecular topology. All right, like my face, if I were to close my eyes and touch my face, I can feel my face, and it feels a certain way. And every person's face is going to have its own unique topology. You can think of an antigen as some protein or something that's being expressed on the surface of something that has a specific molecular topology that can be recognized by other molecules. All right. Specifically antibodies. Antibodies recognize antigens. So there are 2 types of antigens. There's actually, in blood typing, we're going to talk about 2 types. In fact, there are more than 2, but there's specifically here 2 types that are the most commonly referred to. So there's type A, and the way this cartoon depicts it, type A antigen has these red circles. And then there's type B, and type B is depicted as little blue triangles. So at birth, this is probably going to be the only immunology I really talk about all semester. There is this process of thymic deletion is what it's called. So childbirth is amazing. Every year I think about trying to convince them to let me teach a class just about development, fetal development and childbirth, and then the birthing process, and then raising a child, because it's incredible. But so right after a child's born, in utero, they're under the protection of the mother's antibodies and the mother's immune system. But ex utero, postpartum, the baby goes through this process called thymic deletion, where their thymus, which is a gland that lives right on top of the heart in the infant, samples tissue antigens from all over your body. And if a red blood cell has been, actually the way it works is red blood cells circulate all over your body. And if a red blood cell gets activated because it has an antibody that recognizes an antigen that is self, an antigen that is on any cell that's in your body, then that activated T cell or B cell gets deleted from the population of white blood cells in your body. So, because the way the immune system works, these antibodies that are going to recognize all these different molecular topologies, they're not hard-coded. You don't have a single gene that's going to recognize exactly, using my analogy of face, there's not going to be one gene that recognizes just the shape of my face, and then one your face and each person's face. The way it works is you're going to have a gene that recognizes this nose, and there's going to be a gene that recognizes this mouth and these eyes. And what we do is we just juggle them all together. We combine them in all the different ways you can combine them. It's sort of like condensing image data in a JPEG or something like that. It's a little bit like that. So there's a high degree of efficiency in the genes that encode for antibodies. Because of that, though, one of the consequences is we make antibodies that recognize the self, right? And you don't want those because that's an autoimmune reaction that you don't want. So we got to get rid of those T cells, all right? That's called thymic deletion. So in a baby that has type A blood, their red blood cells have this red circle antigen on the surface. And all of the antibodies that recognize that red circle get killed off in the first week of life. They get activated and the thymus kills them off. No problem. Yeah? This is the hemolytic syndrome of the newborn that I'm going to talk about in a few slides. So just hold on to that thought. Very good question. So in a person with type A blood, they have they have antigens for A, and they have antibodies for B, because B never got killed off, right? The T cells and B cells that make these antibodies never got killed off. They didn't need to during thymic deletion. And vice versa, a person with type B blood, they have the triangles, but they have the antibodies that recognize the circles. Well, if you have type AB blood, you express both antigens, and both antibodies get killed off. And if you have type O blood, you don't express either antibody or antigen, and you have both antibodies. Okay, so if you take some type B blood and you put it into a type A person, you're going to have antigens that are going to come in contact with antibodies that recognize them, and it's going to bust up those cells and kill them. All right? This is why you need to give a person with type A blood, they need to be given a blood that does not have the type B antigen. So they can receive Type A can receive type A or type O. This is why type O is considered to be universal donor. O can only receive O. O can only receive O. AB can get it from anybody. They can get it from anybody because they don't have antibodies to any of it. All right, so they're the universal acceptor. Luckily for people with type O blood, that's like 60% of the population, so there's no shortage of type O blood out there. All right, blood typing. Was that redundant to some of you guys? Maybe some of you. Yeah. All right. Recycling of the red blood cell. So I said that, or maybe I didn't say, but red blood cells have a lifespan of like 120 days or something like that. And about 1% of your red blood cells get turned over per day. That's 3 million red blood cells per second. That's pretty incredible. 3 million cells per second are getting broken down and new ones made. So let's look at that. We have red blood cell formation in the marrow of your bones, for example, or your spleen. And some of those are going to spontaneously break down. Some of them are going to spontaneously break down. And that hemoglobin is going to get excreted, a small percentage of 90% of the red blood cells and about 10% of, well, maybe a little less than that, 10% of the cellular fragments are going to be picked up by the macrophages in the liver. These are the big, I show that picture of the macrophage that had all the weird arms coming off of it. I showed that picture. Macrophages, yeah, are the cells that, it's like the macrophage that ate Philadelphia, right? It goes out and surrounds older red blood cells and cellular fragments from the spontaneous breakdown, and is going to recycle all that stuff. So amino acids are going to get transported back to the marrow for repackaging, and then we're going to take the heme out. We're going to take the heme out. Some of that heme is going to get used, and some of it is going to be converted into biliverdin, which gets converted into bilirubin, which will bind to albumin and get shunted to the liver. And at that point, we use bilirubin to help form the bile salts, and it's going to get excreted in the bile and dumped into the colon, which is what gives our feces its distinctive color. It's what gives our feces its brown color. A mixture of biliverdin and bilirubin in the feces is what makes it that color. So that's how it's recycled, about 1% per day. All right, hemostasis. So there is a few phases. The first phase is the vascular phase. So we have a cut happens, some sort of injury happens, and the smooth muscle, that tunica media, the smooth muscle layer, the middle layer of the artery or vein that I talked about, you cut it, and a bunch of chemical cues cause the muscle fibers to just go and contract really hard. So try to pinch off the aperture of the capillary and stop the bleeding a little bit. Then we move to the platelet phase. Various chemicals. So I list some of them there: the ADP, thromboxane, A2, calcium, other platelet factors. It's not important to know what, but there's a bunch of chemicals that activate the platelets and cause platelet aggregation. I was in graduate school at University of Virginia for a while. I did a rotation, a research rotation in this lab that was looking at how cocaine is a chemical that can actually potentiate platelet aggregation, which is one of the reasons that people who are cocaine users have a higher likelihood of thrombotic stroke or heart attack. All right. What else? So I think that's all in the platelet phase. But the platelets basically they become sticky. And then we move to the coagulation phase. And this happens shortly after the injury. You have the platelets are kind of the bricks I told you that start plugging the hole. And then there's an extremely, I have this like little white box that says clotting factors. Maybe there's no shortage of things to make you guys go, wow, that's complicated in this class, I guess. But to put up just like even a rudimentary diagram of the clotting cascade intimidates me, that's for sure. It's a very complicated sequence of chemical events that happen that eventually lead to fibrinogen, this apo enzyme, getting activated and cleaved into fibrin, not apo enzyme, proenzyme, pardon me, zymogen. The word is zymogen. Proenzyme and zymogen. Fibrin comes in and acts as the mortar here that helps plug this. So we can see a picture here. You have some red blood cells. There are going to be some platelets in there, and this fibrin meshwork that kind of like glues it all in place and sort of plugs the hole. All right, on to Amanda's question. So, hemolytic disease of the newborn is a problem with another blood antigen. So there's an antigen called the rhesus factor, or Rh. They call it rhesus because it was first identified in rhesus monkeys. Yeah, you know. So the way this happens is if an Rh negative, meaning a woman who does not have rhesus antigen, has a child with an Rh positive father, then there's the potential for an Rh positive baby in an Rh negative mother. All right, and during delivery, not all delivery proceeds totally smoothly. You can have some kind of problem with the placenta, and as the baby's being delivered, there can be some improper detachment of the placenta and hemorrhaging, where the problem is not that the mother's blood might get into the baby, but that the baby's blood gets absorbed by the mother. Now, it's rare, right? This is pretty rare. But if the mother's blood gets inoculated with a little bit of the baby's blood, the mom can make antibodies to this stuff, right? Because she didn't lose them during thymic deletion. She still has the anti-Rh antibodies, the potential to make them. So she's gotten dosed with some Rh positive antigen, and she starts to produce a bunch of these antibodies to kill off these foreign invaders. She doesn't recognize these blood cells. Second pregnancy comes along, and the baby is Rh again, maybe. Maybe the baby's Rh positive again because of the father. All the fathers. The mother is going to be exporting some of these anti-Rh antibodies into the infant's blood. This is a real problem for that baby. So here's a Do I have a picture? Oh yeah, there's some pictures. Oh, poor girl. Look at that. So you can see this baby is being completely attacked by the mother's immune system here. And there are some signs. You can get a positive Coombs test, which is the slide previous that I sort of slipped by. We can go through it. You can look at bilirubin and cord. They, of course, have this hemolytic anemia. The baby is anemic, meaning a low blood cell count because the mother's immune system has been killing off the fetal hemoglobin. Sometimes they can have, this is obviously a very extreme case here. This baby is otherwise healthy, right? If you can get them through this baby is going to be a normal person. But it's a problem here. So prevention, if you know that this is a possible problem for the mother, you can give them this stuff called ROGAM, which is going to basically take the mother's antibodies out of commission. It's going to bind with these anti-RH antibodies and tie them up and prevent them from interacting with this. I'm going to skip going through the Coombs test exactly how they do that, but you can imagine that there's a direct blood test that they can do. All right, so that's all that. I'm going to move into the heart. I have an hour to tell you how the heart works. So here are the objectives. If you were looking through that portion, these would be the objectives that you may have gleaned. So this whole objective thing is a different way to think about it. Rather than focusing on slide by slide, you're looking kind of from the professor's perspective, like what are the overall topics I want to talk about here, like 5 slides on this topic, 2 slides on that. It's a different way to outline this material, a different way to break the material down. So like if I were testing, for example, I, as a person, would put together a test. And it's not just me, but I'm talking more generally. Perhaps as, because this is how you start a class, right? Well, as a person who's building a class, I'm just going to write out what my objectives are. What do I want to teach the students about? That's kind of like almost a syllabus, right? Just bang, bang, bang. These are the topics I want to talk about, and then you flesh them out into slides. So as you're making a test, I want to probably ask a question about each of these topics. Right? That's how a typical professor would build a class. So it's getting in the professor's mind is a good way to study. Think about how they built the class so you can think about how to study. I'm going to talk about the cardiac cycle. I'll talk about in that I'll talk about the conduction system, how that works. And then once I do that, I'm going to talk about the ECG, the electrocardiogram, as a way an experimental way to reveal this conduction behavior. Then we'll get into cardiodynamics, exactly what's happening, where we're pumping blood this way and that. And then I may or I may not, depending on how much time I have, talk about the autonomic innervation of the heart and how that relates to blood pressure regulation. And then if there's time at the end, which there may not be, I have some really cool images, some of them from Roisin Morgan that I talked about in lab. She gave me a bunch of her pictures. So I can just, we'll talk about some imaging. And at the end, maybe I'll skip the imaging, I don't know, but I have a couple slides about heart disease, and maybe we'll get to that. But important parts for sure are the cardiac cycle and cardiodynamics. We'll cover that for sure. All right. And of course, my history. So here's Galen's depiction of the heart. With Galen, he has the heart, the right chamber receiving blood from the liver via the vena cava. So that's essentially correct. Yeah, Galen knew early on most of the plumbing. He has here this liver, which is the source of what he called natural spirits, giving blood into the right side of the heart via the inferior vena cava. Now, he's got some of the plumbing kind of wrong here. I don't even know what this is. I guess this is the descending aorta, and he's got the ascending aorta, and this goes into nothing. I don't know what this structure is meant to be. This should be not here at all. It should be attached to that. So he's got some of the details wrong. But remember, Galen wasn't able to do careful dissection, right? He had to look at the butchered up gladiators and just kind of peek in there before he packed them up, or he could cut up all the pigs that he wanted, of course. So he has a lot of it pretty well mapped out, but not all. For example, he's got the pulmonary arteries coming out of the right atrium when they should be coming out of what is that? Is that that should be coming out of this side. He's got a lot of this kind of messed up. But actually, he did a lot better than our pal Leonardo da Vinci, who's about 1,300 years later, and who is so known for his careful dissection or his careful illustration. But he's got I don't know, that's a mess up there. I don't really know what he's trying to show. But he has the veins and the blood originating from the liver. He thought the liver was the center of the source of your blood, which is understandable given the fact that a tremendous amount of blood passes through the liver. We'll talk about that in the digestion chapter. All right. So I showed you this picture earlier. And when we were talking about the structure, the microanatomy of the heart wall. So I'm putting it again in here to remind you of the epicardium, the myocardium, the endocardium, and then to show the muscle fiber direction is the point of this slide. So the way the heart works is it doesn't just like contract like this. It sort of rotates down. It squeezes down, almost like wringing all the blood out of those chambers. The way the muscle fibers run is in this sort of spiral that heads towards the apex of the heart. If you actually track the muscle fiber direction, so when the heart contracts, there's this like sort of twisting motion to the heart. That was the point of that. So, again, looking at cardiac muscle cells, we have these intercalated discs that we pointed out. I think I asked about that on one of the lab test. Intercalated disc, and if you look at, so here's one muscle cell, here's another, maybe following this line right here. We see an intercalated disc, which right along here. This is a desmosome, okay? But the line gets broken a little bit. There's this little path here. This doesn't follow like a nice clean line between the two muscle cells. This horizontal region we see right here, this is where we have the positioning of a bunch of gap junctions. Gap junctions, you'll remember, are like the grommets in a tarp or whatever, and it allows there to be what's called syncytium. So within cardiac muscle cells, there is kind of a single coherent cytoplasm that runs throughout all the large tracts of muscle tissue. And what this does is it enables, rather than having just like one large muscle fiber, I said you can have muscle fibers that are 30 centimeters long or whatever in your skeletal muscle system. Instead, large portions of the heart are acting as one. All those muscle fibers, even though they are individual cells, because they're all buttoned together this way with the gap junctions, they often act as one complete unit. All right. All right, so to remind you of this anatomy and then point out this single point here. So there's the SA node, internodal fibers, the AV node, the AV bundle branches, moderator band, and Purkinje fibers. So we're going to talk about how this whole system works, this conduction system works. Once the signal gets to the AV node, that's the last real domino that needs to fall. And after you get here, everything else that happens is super fast. So about 1 to 4 milliseconds from when this turns on to when all of this begins to be lit up. All right, so it's extremely fast conduction through the heart. All right, so to begin to talk about conduction and lay that on top of this template of cardiodynamics, we're going to think about the cardiac cycle. So the cycle that a heart is going through, all right, the contractive cycle that the heart has to go through. And we have a cardiac contraction that is going to be mapped by these ECG. ECG is going to give us a look into all of the different electrical phenomena. So we have a contraction. It's what gives us our heart sounds. This contraction is going to pump the blood, which is going to keep our blood pressure up. And then we're going to go into cardiac relaxation and come back around for the next cycle. Beginning of that cycle, then the electrical stimulus starts in the SA node, gets distributed down to the AV node, and then gets distributed throughout the heart. Triggers another contraction cycle. All right. So maybe put a little start right there. That's kind of where we start. An electrical stimulus at the SA node gets distributed amongst the conduction system. Yeah, I should put a little start right there. All right, so let's look at the membrane potential in the SA node, the fibers of the SA node. So turns out in the heart that the threshold is at minus 40 millivolts. So let's just start right here. We're going to start, we've reached threshold, and we have the muscle is going to, the transmembrane potential is going to depolarize, and the muscle is going to contract. The way in this profile is just the same as the transmembrane profile we've seen in things like neurons. So then this is where the sodium's rushing in, sodium gates closed, potassium repolarizes and brings us down here. The thing about these sinoatrial nodes is it doesn't just maintain at the resting potential. There is this passive depolarization that happens. And that's because well, maybe you can guess why you think that is. The spontaneous depolarization. It's related to the expression profiles of the specific sodium-potassium ATPase and the leak channels, those expression patterns, relative expression patterns that are happening in the membrane. So the way this gets tweaked is such that this membrane is going to just passively depolarize and bring itself up to threshold quicker. When that happens, so let's walk through this step by step. Step one, when that happens, the sinoatrial node is going to begin to depolarize. And this is going to be what sets off atrial activation to begin with. So SA node has turned on, and it spreads out across the surface of the heart and is going to begin to cause the muscle fibers in the atria to contract. They do contract, and in so doing, they're going to force blood out of the atria into the ventricles. And meanwhile, that's going to trigger a depolarization in the AV node. So from the beginning, SA node to AV node, that's about 100 millisecond delay. All right. After atrial contraction is well underway and the AV node has been tripped, a signal gets sent down along the bundle branches and through the moderator band. And we're on the cusp now of a contraction in the ventricles. So eventually, all of the muscle fibers in the ventricles are going to contract. And when that happens, the atrial contraction is done, and we're getting contraction here. Pressure is going to rise in the ventricles, and these doors are going to slam shut. All right. So the pressure is going to increase there, and eventually it's going to blast the blood out of the ventricles. But that's the end of the conduction cycle right there. So let's see how this is depicted by the electrocardiogram. When you look at an ECG, it was formerly called an EKG because the technique was developed in Germany, and cardio starts with a K in German. So there's this P wave and a QRS complex, and then a T wave. That's what you see on an ECG. P wave, QRS, T. The P wave is so the way you do it is you stick all these things, these electrodes over the patient, and you're going to pick up all this electrical activity and synthesize it into this trace. And this is time along here, and it's voltage in this direction. So this first bump is the electric field that's being generated by atrial contraction. QRS complex is similarly the electric field that's being picked up by ventricular contraction, whereas this T wave is the repolarization of the ventricles. There probably is a little bit of a bump like this when we're getting repolarization of the atrium, but it gets swallowed up by that QRS complex. All right. So to go further, we need to talk a little bit about specifically the action potential that happens in a cardiac muscle. So here we go at here's some sort of stimulus. It's going to depolarize, and we reach threshold, and there's this depolarization, which is rapid, where sodium is flooding the myocardiocyte, the cardiac muscle cell. And this only takes 3 to 5 milliseconds, very, very short duration. And as soon as we get up to, around plus 30 millivolts, the sodium doors close, and we reach what's called a plateau. This is where we diverge from skeletal muscle. We get to this plateau, and it's because there is the calcium floods in, these calcium gates flood in, but the closure of these calcium channels is very slow. So we're not able to sequester calcium as rapidly as you can in the skeletal. And it gives us this plateau. where we're stretching out this absolute refractory. It's just way high potential, and we kind of dwell up there for a while. We dwell in this absolute refractory period for much longer than a skeletal. So if you look at skeletal muscle, this red thing here, it's on the order of tens of milliseconds. But we are in this absolute refractory period for 200 milliseconds in a skeletal muscle. Much longer time. And we'll talk about that. Well, I'm going to make you tell me why you think that's the case in a bit. And then we begin to repolarize. The potassium channels are opened, and potassium helps to repolarize the membrane. We get back down, emerge from the relative refractory period after 250 milliseconds or so. So does anyone want to guess why we have this long refractory period? Yeah. Certainly isn't good for you. Yeah, why? I'm going to give you a clue. Often when I ask these questions, I put the answer on the board. And if you just look at the board, you'll get the answer. It prevents wave summation and tetany, right? You don't want your heart to go into tetanus, meaning you don't want the heart muscle to just be like, you want the heart to keep going through its cycle, keep going through the cycle. Because if it goes into tetany, it's not going to be able to actually fill with blood and then release. You need it to relax so that the heart can fill with more blood, so that you can pump more blood out. If it was just in tetany, then it would be constantly contracted, and you wouldn't be able to fill the heart with blood. So this time period here is a reflection of the minimum time required to get any kind of substantive filling the heart of blood, so that you can pump more blood out. Does that make sense? All right. So that was pretty much the cardiac conduction system and the contraction of a specific neuron. Now I'm going to go through the cardiac cycle, which will lead me to cardiac cardiodynamics. So two words to define for some of you. Systole, S-Y-S-T-O-L-E, is contraction, and diastole is relaxation of the heart. So when the heart is in, it's not the heart, but the ventricle or the atrium is in systole, it's contracting, and when it's in diastole, it's relaxing. So the atrium and the ventricles have different systole and diastole patterns from one another, as you can see here. So let's start at the little yellow box up there. The first step is going to be the beginning of atrial systole. At the beginning of this, right here, during this part, we're in ventricular and atrial diastole. So everybody's chilling out. We get to this point here where the SA node has fired off, and it starts atrial systole. So the atrium is going to contract, and it's going to force whatever last amount of blood into the ventricle that we can get in there. Then the atrial systole will end. When the atrial systole ends, we're going to begin ventricular systole. And ventricular systole leads to the contraction of the ventricles, the increase in the pressure of blood in the ventricles, and then eventually we're going to get to this point where the pressure of blood in the ventricle is higher than the pressure of blood in the aorta or the pulmonary trunk, and higher than your ambient blood pressure. when that happens, the doors will snap open and the blood will get forced out. Pardon me. Hiccups. So eventually that will end, and we'll enter ventricular diastole. So we're in diastole for both right now. And what's happening, we're just waiting to refill the heart with blood for the next contraction. So blood's coming back into the atria, and we begin to get this passive filling of the ventricles. Just whatever the back pressure is, all right, whatever that preload is, whatever the pressure is from the venous blood, whatever that pressure is, it's going to passively fill the ventricles via the atria, all right, until we get back around the wheel. Okay, so if there's one chart on this PowerPoint that it would be wise to appreciate, I think it's this one, because this really integrates everything I've been talking about so far into one picture that perhaps you can begin to appreciate. So along the top is the ECG, the electrocardiogram. So that is really just a way for us to track the electrical phenomenon. We said the P wave is associated with atrial depolarization. QRS is ventricular depolarization. T wave is ventricular repolarization. And the next one is the beige square in the middle. The beige panel in the middle is looking at pressure along the same timeline. So let's just focus, let's not worry about the left atrium so much. I mean, we can do that, I guess. Left atrium, the pressure, we start atrial systole right here. Atrial systole happens and the pressure goes up in the left atrium. We're going to skip the right side. We're just going to talk about the left side. The same pretty much over there. The pressure goes up in the left atrium and it's forcing blood into the left ventricle. The left ventricle's pressure matches that of the left atrium. All right? Until we get to the end of atrial systole and the atrium closes off. All right? And we begin ventricular systole. Well, the ventricle starts to contract. And so at this point, the ventricle and the atrium are separate chambers, and they're going to have different pressure profiles. Here, the door is open between them, but then the door shuts, the left AV valve closes, the tricuspid valve, bicuspid valve, we're on the left side, this is the mitral valve, pardon me. The pressure in the left ventricle starts to go up. And this is because the volume is not really changing that much. And this is volume right here. You can see from this point to this point, the volume is not changing at all. And since the volume's not changing, but the ventricle is contracting, that pressure skyrockets. All right, pressure skyrockets until we get to this point right here. It's just this far down the line, just a little bit down the line. Just going from here to here. At this point, the blood pressure in the left ventricle equals the blood pressure in the aorta. These are the same. The blood pressure is the same. And when this happens, as the ventricle continues to contract, it's able to overwhelm the pressure in the aorta and push the doors open. Push that aortic semilunar valve open and eject blood from the ventricle into the aorta. You guys following this? Pretty straightforward. So at this point, the aorta and the ventricle are the same. They're open compartments, right? So they should have the same pressure profile. And the contraction goes up until we reach this point where the ventricle is kind of squeezing the last of it out. It can't continue to contract a whole lot more, all right? And the pressure starts to wane until we get to this point where the aorta, the capacity of the aorta to maintain pressure exceeds that of the ventricle, and the aortic semilunar valve closes. All right, and then the pressure wave in the aorta does its thing, and pressure starts dropping off. All right, and we can see this in the volume here. So this is the volume of the left ventricle. It's passively filling. Then the atrium contracts, and we get this active filling until the end. And then this is the max volume. This is what's called the end diastolic volume, And then we get contraction here until the doors open up, and we eject all this blood until we get to this point right here, where we reach end systolic volume. All right, and then this is the beginning of another cycle where we get passive filling. So we're in ventricular diastole right here, and we get to passively fill with blood once again. So the difference between end diastolic volume and end systolic volume is going to turn out to be important. We call this volume stroke volume, and I'm going to introduce that again in another couple slides in a different way. But it's important because the larger your stroke volume, the more blood that you're pushing into your system per beat, right? So that's going to be 1/2 of something called your cardiac output. If you take your stroke volume and you multiply it by your heart rate, how many beats you're getting per minute, that's your overall cardiac output. All right, that's giving you a sense of how much blood your heart is able to move. And there'll be a slide where I go through it in its place, but I'm just doing that while you're seeing this on the slide here. I think that's everything. I could go into a little more detail, but I think you get it. Oh, wait, important. These two pressures, the preload and the afterload. So those are important concepts. The afterload is the pressure against which the heart must work to eject blood during systole. So your afterload is essentially the pressure of the blood in the aorta. All right, it's the pressure of the blood in your aorta leading into systole. It's the pressure that the heart has to achieve before it can snap those valves open. So think of it like this. If the afterload is lower, if you have a lower pressure in the aorta, then the valve is going to snap open here, and you'll be able to eject more blood. You'll be able to increase your cardiac output. So people with lower blood pressure, lower arterial blood pressure, are going to be able to move more blood per stroke. They're going to be able to move more blood per heartbeat because they don't have to work against as much resistance in the aorta. Does that make sense? So there's the afterload. This is one of the factors that we're going to be able to affect the cardiac output is by changing afterload. Because afterload is directly related to it's going to be directly related to actually end systolic volume and thereby stroke volume. All right, and then there's preload. So this is the end volumetric pressure that is going to stretch the ventricle. How high is the pressure on this end that's filling the ventricle? So the higher this pressure goes, presumably the higher your end diastolic volume. This is also going to affect stroke volume. So things that will positively impact cardiac output are going to be a lower afterload or pressure in the aorta, or a larger preload, or how much blood you can fit into the heart initially. All right, so I'm going to go through, that's called cardiodynamics. I'm going to go through those in detail in kind of a different chart, but I'm just showing it to you here, introducing these concepts here. And then I couldn't fit this quite on the bottom of that slide. I could have had another banner along the bottom that just show the heart sounds, the lub-dub. There are 4 different heart sounds. 2 of them are the ones that are commonly heard by people. If you are a doctor, you're trained to listen for them all. But yeah, so the lub is the semilunar valve opening. It's that aortic semilunar valve opening up. It's large. And then the dub is just the opposite. It's just the semilunar valve closing. Just the lub and the dub. Here's a little chart that, I don't know, it's probably not even necessary to have it in here, but it's just showing the different places on the chest where you can put your stethoscope and what they're going to report on, right? So you can if you want to know what the aortic valve sounds like, you can place the stethoscope here, or you want the pulmonary valve to be here, for example, et cetera, et cetera. It's actually not that important to this conversation. All right. So I've already said all of this here, but I'm going to go through it again because it's important. So just to define these terms, the end diastolic volume, this is how much blood is in the heart before at the end of diastole, before we enter systole, all right? So this is how much blood we have to work with. The higher the preload, the higher this value is going to be, all right? The higher a pressure the atrium can create, or the higher the venous pressure that's helping to fill the blood, fill the heart, the higher this end diastolic volume. Then there's the end systolic volume. That's the amount of blood that's left in the heart. And this is a function of afterload. If your afterload is really high, if the pressure in your aorta is really high, then this is going to be a higher number, and you're going to have ejected less blood. Does that make sense? Yeah? Just shake your head if it doesn't. Okay, so that's good. Stroke volume is the difference between those. It's just the difference. It's how much blood per stroke is getting ejected. So in any kind of pump, there's going to be some amount of material that hasn't been fully ejected. There's still some left in the pump, right? There's some blood always left in the heart. The heart doesn't squeeze every single drop out in a beat. It takes it down to this end systolic volume. It gets rid of most of it. So the difference of those two is your stroke volume. And you can think about that as milliliters per beat. How many milliliters are ejected per beat? The ejection fraction is a number that cardiologists talk about, and it's the percentage of your end diastolic volume that is getting ejected in stroke volume. So if this is 100%, what percentage does this amount represent? I already gave the definition of cardiac output earlier. It's just the volume pumped by your left ventricle in a minute. So to find that out, we take milliliters per beat and we multiply it by beats per minute. Beats cancel out and we get milliliters per minute. It's just heart rate times stroke volume. So the rest of cardiodynamics is essentially talking about ways in which we can maximize cardiac output. Factors that affect cardiac output. So in general, there's going to be two ways you can affect cardiac output. You can affect your heart rate, or you can affect your stroke volume. Does that make sense? All right. So we're going to talk about different ways of affecting your heart rate and your stroke volume. Am I going too fast for anybody? Any questions, any mysteries? This is clear? All right. Right, what I just said. So cardiac output is heart rate times stroke volume. And these are the ways that we can affect heart rate, and these are the ways we can affect stroke volume. So let's start with heart rate. The two ways we can affect heart rate are autonomic innervation. So our autonomic nervous system does have an influence on our heart rate. I didn't get a chance to talk about autonomics the other day because I was not rushing, but autonomics are basically the sympathetic and parasympathetic nervous system. And then the next way is there can be different hormones that can be released. Epinephrine, for example, is going to have an impact on your. Is it that movie Pulp Fiction where Uma Thurman goes into, is it Uma Thurman? I don't remember who. She goes into cardiac arrest, and then like John Travolta so dramatically just takes this giant hypodermic and jams it into her chest and pumps her full of epinephrine. No. And then she gets up, and I don't really remember, but it seemed like she was fresh as a daisy. On the other side, factors affecting stroke volume. End diastolic volume and end systolic volume. I mean, because that's how we calculate stroke volume. So I've already talked about afterload and preload. I'm going to go through that in more detail. I'm going to take this table or this chart and I'm going to blow it up. And you're going to see a more complicated version in just a moment. But let's start going through them a bit at a time. Here is the autonomics. So we're going to work our way across this table. Autonomic, hormone, end diastolic, and systolic. First, in the autonomic innervation, there can be sympathetic or parasympathetic. The sympathetic nerves are, so there's this, I guess the place to start is in the medulla. In the medulla oblongata, there are two centers. One is the cardio-acceleratory center, and the other is the cardio-inhibitory center. The cardio-acceleratory center is sympathetic in nature, and the cardio-inhibitory center is parasympathetic in nature. So first, the sympathetic. The sympathetic fibers descend the spinal cord and emerge along. These are motor fibers, so they're coming out the ventral root. Because remember, the dorsal root is sensory, ventral root is motor. They come out the motor root and then go and synapse in this ganglia, part of the sympathetic chain ganglia. The sympathetic chain ganglia, I didn't get a chance to talk about, but it's this chain of ganglia that runs parallel to your spinal cord. For any of those of you who are lucky enough to go to a cadaver lab, it's usually not on the list of things they make you look for in the cadaver lab, but it is definitely worth looking for. Once you've fully excavated the chest and taken everything out of there, it's worth digging around along the spine back there, carefully digging, to reveal the sympathetic chain ganglia. It's pretty cool. So these sympathetic nerves, I told you that autonomics are going to have some sort of synapse. There's a preganglionic fiber and a postganglionic fiber. I told you that the other day. The synapse happens in this ganglia, and then we have this postganglionic fiber, which we call the cardiac nerve. It's going to go down to the heart. and it's going to synapse on the SA and the AV node and go throughout. It's going to speed up this process. It's going to speed up the contractions of the heart, specifically by affecting the pre-potential of the heart, that the distance between the resting potential and the threshold. It's going to change that so that the heart reaches threshold quicker. On the other hand, there is the parasympathetic innervation of the heart, and this comes out of the medulla oblongata, comes out of the brainstem via the 10th cranial nerve, the vagus nerve. Vagus nerve does all kinds of things. It is the vagrant of the body, vagus meaning vagrant. It affects the lungs, it affects the heart, it affects the GI tract, it does other stuff. But it's the vagus nerve that comes down here and synapses on the heart and is going to affect the heart rate in this equal and opposite way to the sympathetic. It's going to change that pre-potential so that the resting potential of a myocardiocyte is further away from threshold. All right, and it takes longer for that cell to depolarize to threshold passively. Does that make sense? Okay. Autonomics. So here this is same thing in terms of the membrane. So on the top, this is normal, your normal heart rate. You're maybe at 60. The threshold there, they have the threshold at whatever, minus 40, I guess, is the threshold for the heart. And there's this like passive depolarization until you reach threshold. Then there's contraction, refractory period, repolarization, and then passive depolarization. So let's go to parasympathetic. Parasympathetic first, the rest and digest. What this is going to do is it's going to lower it's going to affect this pre-potential. It's going to make the resting potential lower, more hyperpolarized. It's going to hyperpolarize these cells. And in so doing, the passive depolarization that happens is going to last longer. So it's going to spread out the heartbeats. It's going to spread out in time, the depolarization events. Whereas and we call this bradycardia. Bradycardia. Tom Brady, the guy for the Patriots, is that right? Yeah. So I'm in Patriots country here. Just imagine Brady, cool as a cucumber out there, totally just resting in the pocket, right? Slow heartbeat. So bradycardia. It's the first time I've ever done that memory trick there. Woo! Speeding of the heart by sympathetic innervation is going to increase, or I guess the word is depolarize the resting potential. So a reduced repolarization. It doesn't get as low as it does in the other case here. So the time that it takes to passively depolarize, reach threshold, is shorter, and it pushes these heartbeats closer together, more rapid. All right, we call this tachycardia. Like a tachometer is that thing on your car that shows how fast the engine is going. Just means speed. So speedy heart is tachycardia. Person with tachycardia, their heart is racing. All right. Next, I didn't talk about hormones at all, but there's lots of hormones. I hardly got a chance to talk about neurotransmitters the other day, so we're not going to worry about hormones. But hormones achieve the same thing, just chemically, as opposed to with nerves. Right? It's going to do the same, have the same effect. On the other side, stroke volume. How are we going to affect that? We're going to affect it via end diastolic volume and end systolic volume. Let's go through these one at a time. So, what do I have here? Yeah, okay. Let's start on this side. End diastolic volume, right, is a factor. Preload is a factor in determining end diastolic volume. The preload is the pressure in the venous return. It's the pressure that the atrium is able to generate. It's going to affect how much blood is able to be fit in the heart at the beginning of contraction. So at the end of diastole, what's that volume? It's a function of preload. The larger the preload, the larger the end diastolic volume. The larger the end diastolic volume, the larger the stroke volume. So increased end diastolic volume, increased stroke volume. Increased preload, increased diastolic volume, stroke volume. So there are two factors that are going to influence preload, and that's filling time and venous return. Let's do filling time first. The longer you let the heart fill, the larger the volume is going to be. That should make sense. This is why myocardiocytes have such a long refractory period, as Jonathan pointed out for us. So the longer you allow the heart to fill, the more volume is going to be there. The higher that volume, the higher your end diastolic volume, the higher the stroke volume. And then the higher the cardiac output. Of course, the converse is true. So then on the other hand, there's venous return. Venous return is how much blood is coming back from the venous system. All right, how much blood is coming back from the venous system? The more blood that is being returned, the higher your end diastolic volume. The lower that venous return, the lower the end diastolic volume. Low end diastolic volume is low stroke volume, is low cardiac output. My throat is getting a little sore here. I'm going to orange in front of you. I apologize for eating in front of you. All right, so that's end diastolic volume. Let's consider the other side, end systolic volume. So, end systolic volume can be affected by two things. The contractility of the muscle cells and the afterload. So let's think about afterload first, because afterload is just the mirror of the preload. Afterload can be affected by increased vasoconstriction or decreased by vasodilation. All right. So that means this is the pressure against which the heart is working in the aorta. Afterload is the pressure that's in the aorta that the heart is working against. So if it's a higher pressure, you're going to have if it's a higher pressure, you're going to have a higher end systolic volume, which means you've ejected less blood, which means a lower stroke volume. So end systolic volume is inversely correlated with stroke volume, right? The higher your end systolic volume, the lower your stroke volume. This is how much blood you left on the table that you didn't eject. So the more you leave on the table, the less was able to be part of your cardiac output. Increased vasoconstriction. Now, I'll have to tell you, when you're thinking about this, I do not memorize increased vasoconstriction equals this, that with afterload and insight. Like, I don't memorize them as individual facts. If I have to think about it, if you ask me what increased vasoconstriction did, I would just try to make sense of it. Try to think about it logically. All right. So try to understand this whole system in a logical sense, and you'll be able to at any point derive what that's how I would approach this, because a bunch of isolated relationships would drive me insane. I couldn't think of it. So increased vasoconstriction, what is that doing? That's reducing the volume of the circulatory system, right? That's going to increase your blood pressure, and that's going to create more resistance that the heart has to work against, which means the heart's going to be able to pump out less blood, all right? As it pumps out less blood, that's going to increase end-systolic volume. It's going to decrease stroke volume and decrease cardiac output. So basically, higher blood pressure, vasoconstriction means higher blood pressure, means your heart's able to move less blood. And then the opposite is true. So decreased vasodilation. I'm sorry. If you have vasodilation, your blood vessels are dilating. You're increasing the volume of your circulatory system, and in so doing, you are lowering the pressure against which the heart has to work, so the heart can pump more blood out into the aorta. If you're pumping more blood, you're lowering end systolic volume and increasing stroke volume, because more blood has gotten pumped out. If you increase stroke volume, you're increasing your cardiac output. All that should be pretty intuitive if you think about it. Like, you should be able to write all this out without having to memorize a zillion. I know that I'm going to ask you to do that, but without memorizing individual facts. Okay, on this side, contractility can also affect end systolic volume. How well is your heart muscle able to contract? So increased contractility means you're going to eject more blood and have a lower end systolic volume. And there's different ways we can achieve that. And these ways are essentially the same thing that affects our heart rate. So sympathetic and parasympathetic, your contractility gets increased by sympathetic stimulation, decreased by parasympathetic. And then there are a number of hormones that can get the party started. Epinephrine, norepinephrine, glucagon, thyroid hormones, depending on what you need. All right. I think that's everything. That's a pretty good slide. What do I got here? 5 minutes. This one's not that important. This is really just the same thing I talked about on that other slide, but another way of looking at it. I'd rather than going through the same thing, I'd rather talk about something new. Okay, so let's talk about blood pressure just a little bit. Is this what I want to talk about? You want to talk about blood pressure, or you want to see some cool pictures? Yeah. Let's look at some cool pictures. Yeah, just get your dents in here. I don't want to talk about all this. All right. So this is the cardiac cath lab at Maine General in Augusta. And this is where they do all of their imaging. It's pretty cool. I've never been there, but so they have a patient laid out, and they cannulate them. Usually here in the inguinal region, they'll stick something in there, and then they can visualize all of this right real time, right up there on the screen. So here's a coronary angiogram. Is this? Oops. I thought this was a video. Maybe it's not. Okay. So we have some sort of obstruction on in the artery here. Here's the heart. So this would be the right coronary artery wrapping around the backside of the heart over here. And they can send a wire in. There was an obstruction, and apparently they sent a wire in, and were able to clear that. Let's see if I here. So I talked about the ultrasound, and here are some different ultrasound techniques. These slides need to be cleaned up, don't they? So here, this guy's got some gel that's been smeared on him, and this guy here has got this little ultrasound thing. And he can see the chambers of the heart in real time beating on the screen. Okay, so these are videos. So this is what some of these stress echocardiograms go. Oh, right. One of the ways they use it is they can take these echocardiograms, they can stick you on a treadmill, hook you up to the shopping cart here, and have and look at how the heart is responding in real time to the stress echo. So here are some examples of what it would look like. Come on, media not found. See, this is because I copied it from the folder. That's too bad. Huh, okay. It's not worth finding it right now. But cardiac MRI is certainly a gorgeous technique, very expensive. This is what their thing looks like. This is all going to be pointless then. These videos don't even matter. Yeah, this is pretty cool. Here, this video, there's a growth. This person had some kind of cancer, sternal cancer, when they were a kid. Got like high radiation, and then they got this growth. You can see it right here, kind of on one of the leaves of their AV valve, so that it was like this huge blob going in and out between the ventricle and the atrium on the left side of their heart. When they got older, they got cancer. These MRIs can give you this kind of information here. So functional MRI looks at flow. And normal heart, here's a heart with restricted circulation. All right, so that's enough of all this. The videos aren't going to really work. Oh, that's the crazy thing they cut out of that dude's heart that I couldn't show you the video for, sadly. Here's transesophageal echo, where they stick this thing down the patient's throat. Here she is, she's awake. I can't imagine what that's like, but they stick it in there, and you can see how well positioned the probe is with respect to the heart. It's a crappy picture, but it gives nice images. And here are some videos of that, which I'm sure are not going to, the media is missing for all that. All right, so, this is a reconstructed image of that messed up heart valve that I showed with the goo.